Yes, first, thank you to the organizers for inviting me and inviting me again after I was not able to make it the first time. But thank you all for coming. So I'll just very briefly give you a short CV on me. So I'm originally a molecular biologist, educated at University of Colorado in Boulder, and I studied computer science after that because I got very interested in the Human Genome Project and all the things that I could see were needed to bring value to these genome projects. So I got very interested in bioinformatics, came to DTU, did a PhD in bioinformatics, and then did a postdoc in San Diego, came back to DTU to start a research group. So I have about 17 years of research as a faculty at DTU, and I know some of you from that time as well. So that's a brief background on me. And lately, I've been focused on my own startup that I founded, and some of the technology, of course, came out of the research that my group was doing at DTU. So the talk is in roughly two parts. So first, I'm going to motivate the subject to some as best as I can, and it's going to have a biotech feel, and it's going to talk a little bit about some things you may or may not be aware of in how we create solutions, technical solutions for challenges that we have in society, and some things that may have come to your awareness through popular culture and as you'll see. So the background here is some of the technologies we are seeing today come from these drivers of the technologies. So sequencing DNA has been becoming more and more affordable, as you know, and you can see this graph goes back to 2002, so it's over more than 20 years of technological progress, and we can see the costs have dropped off more than exponentially, in particular here in the early 2000s, mid-2000s. And now, of course, we can sequence a human genome for much less than $1,000. And that's actually was seen as a very difficult task not so many decades ago. But alongside that, alongside the sort of ubiquitous and relatively cheap DNA sequencing that we can also do DNA synthesis. So these are tracking maybe not as fast, of course, as sequencing, but the ability to create de novo DNA sequences. In fact, there are companies out there that are synthesizing whole genomes for microorganisms. There have been projects to re-synthesize the genome of yeast as well, so actually even larger genomes. But nowadays, we can synthesize DNA nearly as effectively as we can sequence it. You can see the costs here are still higher, though. It costs more in materials to do the synthesis. So we can read and write DNA, and we would argue as biotechnologists that our ability to read and encode information in DNA is in fact developing faster than our advances in hardware and integrated circuits. So that is something that I think is still going on. This has been going on for a few years now, and how that can find its way into solutions in everyday life. We're starting to see more and more of that, and I know there are some companies that are offering data, long-term data storage as DNA. That is actually now becoming more viable. That in fact, DNA is a very excellent high-density information storage medium. Where we come from, so basic DNA technologies and genetic engineering, and now synthetic biology came from, and I show you these pictures. This is where we came from as biotechnologists. This cut, copy, paste, where we take pieces cut them out from natural sources, and we reorganize them, and we make new things, new technologies based on this cut and paste of DNA. So that has gotten us far. We have developed lots of drugs. We've solved, in large part, many hard problems with regard to getting cells to make things for us to treat us, and such as diabetes and insulin that we all know is really important for the Danish biotech economy. But today we think about this more as synthetic biology, and with synthetic biology we add on this extra technology of design, that we can design new things that we've never seen in biology before. And of course, many of you are possibly aware that many companies out there are designing primarily protein sequences, new proteins that fold in ways that we've never actually seen in nature. And I'll talk briefly more about that. But the synthetic biology field, from which, where we and this is still coming from primarily a cut, copy, paste approach. We've been able to engineer full pathways into organisms. So here we're making a chemotherapeutic, a very complicated molecule called Taxol, or paclitaxel. Some of you may have heard of this. It's used for treatment of breast and ovarian cancer, and something that I also worked on. My first jobs was to isolate Taxol, and it's in very, very low concentrations in Taxus brevifolia. It's a yew tree, Pacific yew tree. And so now it can be made either semi-synthetically from engineered organisms. Another one is the vinblastine story. This has some connection to DTU. It's from a number of years ago, where they've engineered microorganisms to produce a drug, vinblastine, which, as you can see, is a very complicated molecule. So this is something biology is very good at doing, and we are now able to, in some extent, say we want to make this molecule. What are all the steps we need to engineer into a microorganism to make that molecule? From a few years back as well, this got some attention, and you may have seen this, basically brewing things like THC. Of course, that is technically possible. Does it replace the naturally occurring ways of getting THC? No, because plants tend to be already very good at doing this naturally, but nonetheless, this raised a few alarms saying, okay, now people are going to grow this at home. This maybe was around the time lots of states were legalizing use of marijuana anyway, so we don't hear too much about it today. Another wave that we've seen is that we can engineer organisms to make certain proteins that we can add to food. So this is leg hemoglobin. So it's a plant hemoglobin. It's not actually an animal hemoglobin, but it's a plant hemoglobin that we've been able to add to essentially veggie burgers to make them more meat-like. And sure, many of you have experienced that. As well as things like replacing, instead of sourcing silk from moths, silk moths, we can actually engineer microorganisms to produce those proteins in ways that we can simulate that textile. So this is now stepping into what we would call biomaterials, bio-based materials, which is also an area that's growing. And of course, should be, in principle, far more sustainable than farming these animals. This is more speculative, but there is a company that is trying to revive through de-extinction, so to revive the woolly mammoth. They have some arguments for why this could be important for, say, Siberia and maintaining ecosystems. And that technically, if we can synthesize a yeast genome, it shouldn't be too many years before we are able to partially synthesize higher eukaryotes such as mammals, but I would still say we're still fairly far off. We're in the press very recently. This was from a few days ago, where they were showing they could make a woolly mouse. So this is there's some debate as to whether this is relevant for turning an elephant into a woolly mammoth, but nonetheless, they could show that they have some way of controlling the phenotypes of fur in mammals. So the woolly mouse has been given to us. I will switch a little bit here towards, this is more of a general slide showing that how we do this in practice in not the mouse, but anyway, in when we make proteins in a lab, we typically do this in large tanks. It's a process very much like brewing beer. We do a fermentation in a large tank, and there are many steps upstream, so this is called the upstream, and then also many steps downstream of the fermentation. So this is where sort of the products are made, but then of course with regard to drugs, there are many, many steps downstream that are important, as well as many steps upstream. So the company, our startup, which I'll tell you about, we come in very far upstream, where we apply some AI, and I'll tell you more about that in a few slides. where we design the DNA that goes into the cells, we engineer into the cells, which hopefully you can see we are able to do. And then hopefully we can address many different types of bioproduction problems with what we're doing with our AI. You're probably more familiar with how AI is applied to the concept of protein design. So this was out not so long ago. This large language model applied to go from sequence to structure and function with regard to proteins. And I think one of the things that I will sort of focus in on here is the scale of these models, as you may or may not be aware of, has on the order of 100 billion parameters, and it's trained on about 3 billion proteins. So you may, from a classical AI perspective, we are basically memorizing, to some extent, all the protein structure data that is out there, and then we're able to use that, even though we're very likely overfitting quite a bit, we're still able to use that to present these models with sort of partial prompts of whether it's the sequence structure or function pieces missing, and then it can, of course, give you the most likely prediction as to what amino acids belong in the rest of the sequence. So there's a whole set of companies who are trying to do just this, to engineer novel proteins for all kinds of different applications. This is fairly new, but I won't talk about large language models. We don't use large language models. Large language models, of course, require very large corpuses of data. But Tailwind, we say we are using AI to build intelligent DNA sequences. And first, maybe we'll have to sort of see where does this technology fit in. When we have a genome, DNA sequence, we need to make some things from that DNA, and there are a number of steps that are needed to get from the DNA to the protein, and it's not meant to be a lesson in molecular biology, but this is just to highlight where we are. We're in this step, the transcription step, so when we go from DNA to the RNA, which is this intermediate messenger that then gets translated into protein. But there, of course, are steps of regulation all along the way, but we are mostly concerned with engineering the DNA that gives us the best transcriptional output. And that means we are focusing on promoter sequences, which I'll tell you about in a moment. So the problem here in biomanufacturing-based companies is that engineering their strains for a new bioprocess takes a lot of time. Maybe they don't have the methods to select the best sequences. There's a lot of trial and error that's going on. That's common practice. And there are many suboptimal bioproduction processes that take many years to debottleneck or to optimize. So, like, for example, the initial groups that had yeast making cannabinoids, the yields are so low. It's usually when you first show that you can get an organism to make something, it's not commercially viable. So there's a lot of technology that needs to be brought to bear to bring these bioprocesses to commercially viable state. And that's where we hope we can do that, help companies get faster to a commercially viable bioprocess. So part of how we do this is just an overview of our method. We have a machine learning approach that takes as input genetic data, and this primarily comes from large publicly available genomes. So there are lots of genomes out there that we can use. We are, as a business-to-business company, we talk to the companies and say, what are your challenges? What are your production hosts? So they need to tell us a little bit about what they're doing and what kinds of organisms they use. And then we can go and find publicly available data on related species and extract the DNA. And sometimes we have to annotate these genomes. Many of them are not annotated. So that's part of our process, annotation, collation of data. And then we can build many models for the functional DNA sequences we want to design. So, I mean, that's the way genomes function. There are many sort of cassettes. So each gene has a whole host of functional DNA sequences around them, including or within the coding sequence itself, that make a gene. So we are able to build for the few thousands or 10,000s of genes in an organism, build models for each gene. And at the same time, we're able to identify also through bioinformatics, which of these sequences might be most suitable for the process that a customer is trying to develop. So we have prior gene ranking or prioritization approach at the same time. So then we present candidates to companies, and here's just some of the data we have from our initial studies. So in the black bars, we're seeing protein production as measured by fluorescent proteins, driven by the expression of our design promoter in dark and relative to a native promoter in the cyan. So we were able to generate designs that outperform the natural sequences, and these are known to be strong. So fairly commonly used, very strong promoter, strong constitutive promoter, it said, so it should be on all the time, producing a lot of transcripts, a lot of gene expression, a lot of protein. So we're measuring protein and comparing to the natural promoters that generate that protein. We've also moved into, this is Comictilla, it's a yeast, and we can see that we have achieved anywhere from 50 to 400-fold, 400% increases over other strong promoters. And this is a very strong promoter in this organism, and we could improve that about 40% or so. So we have shown that our technology works in a number of organisms, including Saccharomyces cerevisiae, which is the standard budding yeast. I don't show you that data here, but these organisms are filamentous fungi that are important to novonesis, and a lot of the enzyme producers use these organisms for production. Our team, we're a small team. We have our CEO, is Mirko, and he's comes with a business background from CBS at KU, and Lorenzo also has a background from Novo and Stanford, and he's helping make usable code out of my academic code, which is very, very good, and doing a very professional job in making us scalable. And we have Professor Henrik Simonsen. He was also at DTU, and he's our plant synthetic biologist, and he's helping us with business development. And of course, I'd just like to thank DTU for helping us get off the ground as a startup, and as well Innovation Fonden and the Danish Agency for Higher Education and Science for support. So I'm happy to take questions if you have any. Thanks.
Thank you for a wonderful talk. Let's see who have questions. Wow, we have an excellent audience today. Yes.
Thank you so much. I'm a little bit curious about some of the, you said that many of the products that are produced through fermentation are not readily usable in the first couple of trials. And I would like to hear more about which issues you're actually facing and how you're going to solve those.
Yeah, so many times people have an interesting problem. They find some technical solution for that problem, but it's not commercially viable for the reason that the yields are too low or their costs relative to what they could sell is too high. So that usually happens when you are engineering a bioprocess. So the first number of iterations, it won't be viable for reasons that you're trying to make the cells do something they're not naturally interested in doing. It's not a benefit to them to make what you want them to make. So it's about engineering clever things like what Enduro's doing, circuits that addict the organism to the products it's making. You trick them into producing more of what you want them to produce. Also, there are lots of competing pathways that might be taking, so if you have 6 steps in a pathway and some intermediate concentrations you want to be rather high are getting siphoned off into other competing pathways, so you would like to downregulate those pathways. So it's like, think about plumbing and valves, and you have flows through these networks, and you'd like to optimize the flow to your product. So there's some aspect of engineering, genetic engineering the organism to maximize the flux of flows into the products you want. Sometimes the products make the cells sick that you're getting them to make. That often happens, so you can use adaptive evolution to maybe also allow the organism to learn how to produce high concentrations of a product that would otherwise inhibit its growth. Also getting the product out of the cells, so the export of the product, export of the protein can also be a bottleneck, a challenge. Does that partially?
Yeah. The last one covered functionality a little bit. Are you covering functionality in any other aspects? The functionality of the product?
We are not. So we are focused on and maybe I haven't made that entirely clear enough in my talk, but we're functioning on the regulatory sequences that control the output of the genes that's like the first step in getting to the protein. Can be a rate limiting step that if you don't get the mRNA made, you don't get the protein. So we're focused on pushing up the levels of mRNA higher, and then what you saw from the bar charts was that we actually could do that and get the protein levels higher as well. So, yeah.
Thank you.
Show of hands, who else have a question? Yeah, coming behind you.
Thank you. I think as a follow-up question to that one, why are you only interested in the promoter sequence and the regulatory sequences than the gene or enzyme sequence itself? Because I think that's where it's like more, more popular, let's say.
It's certainly more popular to focus, and when we are going to have DNA synthesized, the assumption is you will be synthesizing a coding sequence. So we're not doing that. Part of it is we're trying to meet a need that isn't well met. So there are not many companies out there doing exactly what we do. We had the technology to meet this need that we developed at DTU. So that's part of the reason we're trying to focus. When we started on this journey, I was thinking early on, I want to do this, all these things. And immediately someone said, nope, just focus on this. And as an academic, that's a little bit hard to do, I have to say. I would have loved to. And we do offer codon optimization. We can do that. Yes, we can do codon optimization for sure. Designing new protein sequences, that's not something we have focused on. But there are many companies that do that. So I think it's a highly competitive field right now, I would say.
Thank you.
Do we have more questions? Any online, perhaps?
Thank you, and that's a question from Ruth Castro Silva. And she's asking, or she's saying that there's been some concern around DNA data sourcing. So with the AI added to this equation, do you have any concern about the future of DNA AI and how it will power research?
So if I understand the question the synthesis companies also screen to make sure you're not sending in some pathogenic sequence that you're not going to try to make, reconstruct the poliovirus or things like this. So if that's the nature of the question, we are not using any coding sequences in what we build our models on. So by definition, the sequences we use and the sequences we generate can't be used they can be used for lots of things, but they themselves are not, could not be seen as a biohazard. That makes sense. So they should be within the safe practice guidelines. I hope that answers their question.
I hope so too.
But the companies are, of course, very interested in making sure they screen and not accept in their synthesis pipelines anything that can be used for dual use. Yeah.
And if it doesn't answer the question, Ruth, you're welcome to write something again and clarify your question. Any other questions in the room? Yeah.
I have one question myself, because now we are at DTU Skylab, where we are supposed, or looking very much into how we can foster innovation and make more startup or researchers becoming startups spinning out of the university. And you have been on this journey yourself, so I'm a little bit curious to hear about how that has been to go from research and into working in a company. And if you ever dream of going back.
Yeah, actually, it was fairly easy after 17 years. I think having a change in your career focus is not a bad idea. So from that perspective, it gave me something fresh and new to a way of thinking about problems I was approaching. The process at DTU they've been very supportive. So the mentor program we were in, and we were also here as a startup at Skylab. So that was it's all been made it easy for us. But it's hard to run a research group and build a startup company unless you really have someone take full charge of the project outside of DTU. That part, I think it would be hard. Not that I've tried to do both. Well, maybe we did. I tried to do both for a while, and we didn't really launch the company. Is basically what happened. Yeah, too busy.
Do we have another question in the room? Otherwise, I have one. I'm curious about your next steps in Tailwind Biotech. What do you need next? Do you need customers? What kind of profiles should they have? Do you need investors? What's your next big challenge?
Yeah, we've been speaking to some companies of very different sizes, some very big companies as well. We have some customers have been small companies so far, and companies that are developing a new process, and ideally in yeast or filamentous fungi, because that's where we've really proven our technology. We do have a lot of companies approaching us about engineering bacteria, bacillus or E. coli. I'm sure we could do something, but the space is fairly saturated for DNA sequences and what they can use. But nonetheless, we can do that. Some big companies like to do everything in-house, so they don't believe they need us, which is a bit of a shame. But anyway, we're looking for companies that are on the medium size who are developing new technologies and are willing to test out, try new cutting edge technologies. So
cool. Well, thank you so much. I think this was a wonderful. We have one more. We'll squeeze it in.
I just thought of this. Like, I was wondering, like, your product line. So, are you developing promoters that are organism specific, meaning that you only are limited by that organism, or is it depending on the sequence? Like, is it like specific to the DNA sequence to optimize for that production? And I ask this because, are you able to reuse the products that you're making, or is it specific to the application every single time?
They should be able to reuse them. I mean, our business model has changed a little bit, where we're now focusing more on selling our designs. But initially, we would like to license out to many customers the same designs, the same promoter sequences, and they tend to be, I wouldn't say, they tend to be genus specific. So it should work within the genus. And usually a genus there are not that many production organisms from Aspergillus might be one exception. There are 200 genomes for Aspergillus that we know. And but there's a small handful of Aspergillus organisms that are used. And so our design should work in Aspergillus in general. So somewhat specific, but not only one organism.
Can we get to... 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 Can we get to...
That's a good question. That's a very good question. Yeah.
So I was thinking, I'm coming from bioinformatics, and so how do you approach, like, do you patent your technology, or do you keep it a secret, or like, because software can be difficult, and I guess you also publish some of your academic work. Okay. So how do you go from that and into a business?
So the method is what's the subject of the patent, and it should be granted here fairly in the next few months in the US and in Europe. So right now it's the method that's being protected. We initially wanted to we can patent the sequences. Part of the challenge is we would like to patent a library of sequences, and patents don't usually work that way. They want you to focus on this idea of unity. So a collection is not something that's easy to show as a unity of invention. So they want you to patent each sequence. So the cost there could get high very, very fast. But we did, right now we're trying to keep the design secret. The customer can choose to patent if they want, if we sell them the design. So it's a little bit like selling the IP, to be honest. So there should be higher prices for exclusive sales of designs relative to licensing to multiple customers. So that's how we're doing it now.
Thank you.
Thank you so much, and for paddling a little bit for us here. I'd love to say thank you to you and welcome to you.